Imagine a massive library. This library holds the instruction manual for building and operating a human being. Each book represents a chapter of our genetic code. This instruction manual is our DNA, our genome. It contains all the information needed to create and maintain life. Genome sequencing is like reading this manual. Scientists decode the sequences to understand the instructions written in our DNA. It allows us to understand the secrets of life itself. By unraveling these sequences, we can see how cells function and how life evolves. Genome sequencing has revolutionized biology and medicine. It has opened new doors to research and treatment. It helps us identify genes responsible for diseases. By pinpointing these genes, we can develop targeted therapies. It helps us understand how different populations evolved. This knowledge sheds light on human history and migration patterns. It even helps us develop personalized medicine. Treatments can be tailored to an individual's genetic makeup, improving efficacy and reducing side effects. There are two main methods for sequencing genomes: hierarchical sequencing and whole genome shotgun sequencing. Each method has its own advantages and challenges. Let's explore these fascinating approaches. Understanding them will give us deeper insights into the complex world of genomics. Imagine trying to solve a giant jigsaw puzzle. The pieces are scattered everywhere, and it seems like an impossible task at first glance. You wouldn't just start putting pieces together randomly. That would be chaotic and inefficient. Instead, you'd probably break the puzzle down into smaller sections first. You might start by sorting the pieces by color or edge pieces. This is the basic idea behind hierarchical sequencing, also known as clone-by-clone sequencing. It's a methodical approach to understanding the complex structure of DNA. First, scientists break the DNA into manageable fragments. These fragments are like the smaller sections of the puzzle. These fragments are cloned, creating multiple copies. This step ensures that there are enough pieces to work with, much like having extra puzzle pieces. These clones are then arranged in a specific order, like chapters in a book. This organization is crucial for making sense of the genetic information. Each fragment is then sequenced individually. This is akin to examining each puzzle piece closely to understand where it fits. Finally, scientists use powerful computers to assemble the sequenced fragments, like putting the puzzle pieces together. The end result is a complete picture of the genome, revealing the intricate details of our genetic blueprint. Imagine shredding that giant jigsaw puzzle into thousands of tiny pieces. Instead of sorting the pieces, you start assembling the puzzle by looking for pieces that fit together. This is the essence of whole genome shotgun sequencing. In this method, the entire genome is fragmented into small pieces. These pieces are sequenced simultaneously. Powerful computers then use overlapping sequences to assemble the entire genome, like finding patterns in a sea of puzzle pieces. Section 4. Comparing the titans, hierarchical versus shotgun sequencing. Both hierarchical and shotgun sequencing have their own strengths and weaknesses. Hierarchical sequencing is like a careful architect, meticulously planning and building a structure step by step. It's more accurate and easier to assemble, especially for complex genomes. Shotgun sequencing is like a bold artist, throwing paint onto a canvas and creating a masterpiece from seeming chaos. It's faster and cheaper but requires more computational power for assembly. Section 5. Impact on the Human Genome Project. The Human Genome Project, a monumental effort to map the entire human genome, wouldn't have been possible without these sequencing technologies. The project initially utilized hierarchical sequencing for its systematic approach. However, the emergence of shotgun sequencing with its speed and cost-effectiveness accelerated the project's completion. The two methods worked in tandem, showcasing the power of combining different approaches in scientific endeavors. Section 6. Advantages and disadvantages, a tale of two methods. Hierarchical sequencing shines when dealing with complex genomes, providing a clearer roadmap for assembly. Its methodical nature reduces errors and ambiguities. However, it's a time-consuming and expensive process. Shotgun sequencing thrives on speed and cost-effectiveness. Its ability to sequence vast amounts of DNA simultaneously is unparalleled. However, it requires immense computational power and can lead to errors in repetitive DNA regions. Section 7. Conclusion: the future of genome sequencing. The development of hierarchical and shotgun sequencing has been revolutionary. These methods have transformed our approach to understanding the genetic code, allowing scientists to decode the complex sequences of DNA with remarkable precision and speed. These methods have given us an unprecedented understanding of the blueprint of life. By breaking down DNA into manageable pieces and then reassembling them, researchers have been able to map entire genomes, revealing the intricate details of genetic information. They have paved the way for personalized medicine, disease diagnosis, and evolutionary studies. Personalized medicine tailors treatments to individual genetic profiles, offering more effective and targeted therapies. In disease diagnosis, early detection and precise identification of genetic disorders have become possible. As technology advances, we can expect even faster, more accurate, and affordable sequencing technologies. Innovations such as AI and machine learning are being integrated into sequencing processes, enhancing the accuracy and efficiency of data analysis. This will usher in a new era of scientific discovery, where the boundaries of what we can achieve with genetic information are continually expanding. The potential applications are vast, from improving agricultural practices to developing new biotechnologies with profound implications for human health. The ability to monitor and predict health outcomes based on genetic data will revolutionize preventive medicine and public health strategies, and our understanding of the natural world. By studying the genomes of various species, we can gain insights into biodiversity, evolution, and the interconnectedness of life on Earth, ultimately contributing to conservation efforts and ecological sustainability.